Hello everyone. Today I'm going to discuss with you the topic that is biomolecules. Okay. So firstly, what is carbohydrates? Okay. In the starting, it was thought that carbohydrates are the compounds formed from carbon and hydrogen. And it was taken that the formula of carbohydrate should be CxH2O(Y). Okay, in the starting it was thought that, but after some time, uh, they thought that a remanose. This is a carbohydrate. This is this doesn't fit in this CxH2O(Y), whereas acetic acid, which is not a carbohydrate, this one is a carbohydrate, but this will not fit in this journal formula CxH2O5. Whereas acetic acid has a formula CH3COOH, but it will fit in this journal formula C2H2O2. Okay, so this will fit in this journal formula, whether it is not a carbohydrate. Okay, although it is not a carbohydrate. Now, after that, there is a journal definition for the carbohydrate, that is optically active polyhydroxyaldehyde or ketones, or the compounds which produce such unit on hydrolysis are called carbohydrates. I can say, suppose polyhydroxyaldehyde and ketone optically active, so there is a need of chiral center. Okay, I am showing you, and this you need to remember. So this is the structure of glucose. Structure of glucose. What is this one? Okay, what is this glucose? What The structure of glucose is this one. So these are the chiral centers. These are the chiral centers here. That's why the compound is optically active. Which compound is optically active? That rotates the plane polarized light. Plane polarized light. Okay, so this is the polyhydroxyaldehyde, polyhydroxyaldehyde, or polyhydroxyketone is called carbohydrates. Carbohydrates. Now, based on that, ketone is also there. Ketone structure is like this. Sorry, this one is OH. I'll show you. H. This one is also optically active compound. Okay, now this is the structure of fructose. I'll explain in the next slides structure of fructose. Okay, now the next thing is, this is the optically active that rotates the plane polarized light, and this is also optically active that rotates the plane polarized light. Rotation. Okay, rotation of the plane polarized light. Polarized light will rotate like this, and this is the center from where the plane polarized light will rotate. Okay, now this is the for the optically active compound. Now the classification of carbohydrates. Carbohydrates can be monosaccharide, monosaccharides. Classification of carbohydrates, monosaccharides, oligosaccharides, polysaccharides. In the starting, this is which produce such units on hydrolysis. I'll tell you why. Monosaccharides are the units which contain only one carbohydrate unit, like glucose, fructose, ribose. These are the monosaccharides. Next one is oligosaccharide, which have two or more two monosaccharide unit, which gave these monosaccharides on hydrolysis. Means after the hydrolysis, you will get the monosaccharides. Okay. For example, sucrose, sucrose, and maltose, lactose. These are the example of monosaccharide. Sucrose is made up of beta D plus glucose, sorry, alpha D plus glucose and beta D minus fructose. Maltose, alpha D plus glucose, alpha D plus glucose, 2 units of alpha D plus glucose. Here the lactose for the galactose and the glucose. After that, what is polysaccharide? Polysaccharide is which gave two or more monosaccharide on hydrolysis saccharides, or more than two monosaccharides after hydrolysis. Okay. For example, uh, starch, which upon hydrolysis give more number of alpha D plus glucose. So these are the this is the classification of carbohydrates. Or next one is monosaccharides. Monosaccharides can be for the aldehyde, you need to use the aldose. For the keto group, you need to use the ketose. Okay, now the next one is the glucose preparation. Before that, if I'll tell you that monosaccharide, for example, 3 carbons are there. Okay, 3 carbons are there. So for the aldehyde, aldotriose. For the ketone, ketotriose. Okay, for 4 carbon, aldotetrose, ketotetrose. For 5 carbon, aldohexose, sorry, aldopentose, ketopentose. 6 carbon, aldohexose, ketohexose. Okay, these are the carbons, and after that you will get you need to use the aldo and keto for the fructose type and the aldehyde type. Okay, now the next one is preparation of glucose. Preparation of glucose. You can prepare glucose from sucrose. Sucrose on upon hydrolysis, in the presence of H plus, you will get glucose plus fructose. And also starch. See, here the sucrose C12H22O11 upon hydrolysis in the presence of acidic solution, you will get glucose C6H12O6 and the fructose C6H12O6 fructose. And then the starch C6H10O5 whole bracket N, means starch is a polymer which is made up of more than one unit. So upon hydrolysis at 393 Kelvin and 2 to 3 atmosphere, you will get the glucose NC6H12O6. So this is the preparation for the glucose. Now the structure of glucose. You want the structure of glucose. Okay. So firstly, what uh, for the structure of glucose? From the molecular mass, I got to know that this will be the formula for the glucose. After that, we tried a number of reactions for detecting what exactly the structure of glucose is. Okay. So here I have, suppose I am using C6H12O6. Okay. I am reducing this with the HI. So CHOH4 CH2 on reduction gives CH3 CH2 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 CH3. So I am getting this straight chain. Okay, straight chain, which provide me the evidence that each carbon, uh, all the six carbons are attached to each other in a straight line. In a straight line means there is no branch carbon. Okay, there is no branch carbon. So all the carbon attached to each other, all carbons attached to each other in a straight chain. Okay, now the next one is to know how that whether there is a presence of carbonyl group or not. For the detection of carbonyl group, I am going to react this molecule with NH2OH and with HCN. Okay, so with NH2OH, it will form this one, C double bond NOH, CHOH4, CH2OH. With HCN, it is going to form cyanohydrin. Cyanohydrin. How it is going to form? So this is oxime, this one is oxime, this one is cyanohydrin. Okay, so oxime, suppose I have any journal formula like ketone or aldehyde or the carbonyl group, I am reacting it with H2O. You need to remove the water molecule and you will attach this N with the double bond carbon, and after that HCN. Here H plus and Cn minus will attack over here because it will act as an electrophile. This will be negative charge. So you will get Cn O negative, and after that it will take up the hydrogen OH and Cn. Okay. So this is the type of reaction. Now the next one is, this one is oxime, this one is cyanohydrin. So I am using the same reaction here. I am using the same reaction here to form the cyanohydrin. Means Cn negative is going to attack on this. Oxygen negative will take up the hydrogen and so on. Now the next one is, I have these reactions tell us that the carbonyl group is present over here. The carbonyl group is present over here. CH double bond NOH, CHCNOH. Now the next one is, I want to ask whether the CHO group or is present or not. CHO group, to detect that CHO group is present, I am oxidizing it with bromine water. Bromine water. Bromine water is a mild oxidizing agent. So it will oxidize only the carbonyl carbon to CHO. Okay. So from this, we got to know that CHO is present over here. After that, if I am using the strong oxidizing agent like HNO3, HNO3, it's a strong oxidizing agent. Now, it will oxidize both the CHO and CH2OH into COOH groups. Okay, so this one is saccharic acid. Saccharic acid. Okay, so you need to remember the name also for this. This is the oxidation. Oxidation means with HNO3. Now, I want to confirm that whether the OH groups present on one carbon or on the different carbon. So to detect that, we can try this with acetic anhydride. Acetic anhydride is this one. How you can make this? Suppose I am having acetic acid CH3COOH plus CH3COOH. Okay, CH3. I'm writing it from the back side, and you will get, after removing water, you will get CH3. Okay, so this is acetic anhydride. Now, at the end, COCH3. This is just to form the bond. After that, CHOCOCH3, and then we are getting this one, CH3, CHOCO, CH3, and at the last CHO, CO, CH3. Means from this I got to know that 5 OH groups are present on different carbons. On different carbons. Now the next one is, so from this we got to know the structure. Okay. Now the next one is how you are going to form the structure. First one is CHO. You need to remember D plus glucose. D plus means if OH on the right side, OH on the last carbon, last chiral carbon, OH is on the right side, then this one is the structure. Then after that COHH, COHH, CHOH. Okay. So this is the chiral center here. Now this is D plus glucose. Okay. After that, if I want to write the structure of fructose, you need to remember this. Okay. The structure of fructose is CH2OH, C double bond O, OH, HOH, HOH, CH2OH. So this one is the structure of fructose. Okay. Now, if I want to tell you uh, this one, the structure of fructose, see this one here and this one here is same in both the glucose and fructose. Okay, I'll show you the another slide, another one, that how you can make the these structures: alpha D plus glucopyranose, beta D plus glucopyranose. Okay. Now D plus is because of this, means here plus is the for the dextro, and it's experimental. You can't say by seeing it. Okay. So this, I am going to show you the pyranose like structure. Pyranose like structure for the glucose. Pyranose and furanose like structure for the fructose. Okay. So I have this oxygen lone pair is going to attack on this carbon. Okay. Oxygen lone pair is going to attack on this carbon, and after that, how you can do this? Now I'll show you in the another slide. CHO, CHOH, OH, H, CHOH, CHOH, and CH2OH. Now this oxygen lone pair is going to attack on this carbon. Okay, it will attack, actually this carbonyl carbon is planar in structure. Okay, planar. So this oxygen lone pair can attack from the above side and from the below side to form two structures, two different structures. How you can make this? We have oxygen. Okay, we have carbon. Oxygen can attack from this side. Okay, or it can attack from the back side. Sorry, this one is OH. OH. Oxygen can attack from this side, and also it can attack from the back side. So you will have two type of structures. I'm going to show you this again. OH over here, H over here, connecting oxygen with this. Next one is OH over here, H over here, connecting this. We have CHOH. These are the reactions of aldehyde that you already know during carb aldehyde and ketone. Okay. So this is for the, now oxygen can attack from both sides. Now you can see that here I have OH, here I have OH in this direction. If the OH is on the right direction, then it's alpha D plus glucose. Okay. And if the OH is on the left side, then this one is beta D plus glucose. Now remember that the aldehydic carbon is the anomeric carbon. So this carbon is anomeric carbon. Okay, where anomeric carbon, where the hydroxide side is changed. Okay. Now, if I want to make the pyranose-like structure, you just need to make a six-membered ring. Okay, this is your six-membered ring. So, from that, you need to know if the OH is on the right side. You need to remember this trick, that is write down, or if OH is present in the right side, you need to put the OH on the down position. So this is the up position, this one is the down. Up, down, up, down, up, down, up, down. Before I'll start, you need to put the OH and H on this. So OH is on the right side, OH you need to put here. H over here. H upper, then OH here, OH, H, H, OH. Right group on the down position. Left group on the up position. CH2OH you always need to put here and H here. Okay. So this is alpha D plus glucopyranose. Alpha D plus glucose pyranose. So this uh, is equal to the pyran structure. These are the Harvard structure that I am putting here. Okay. These are the Harvard structure. Now, the next one is here. There is some mistake in the structures, but I am showing you. Okay. Now, if you got to know regarding these structures, then how we can make this structure? Again, form the pyran ring and OH on the upside, H on the downside. H on the upside, OH on the downside, OH on the upside, H on the downside, H on the upside, OH on the downside, and CH2OH always up and H. This is beta D plus glucopyranose. So this is the structure for beta D plus glucopyranol. Now the next one is fructose. Fructose. Fructose is CH2OH, C double bond O, C OH H, CHOH, CHOH, CH2OH. Again, the oxygen of fifth carbon, okay, is going to attack on this carbonyl carbon because this is electrophilic in nature, this is nucleophilic in nature, and carbon cannot form more than 4 bonds. That's why it will dissociate. Okay. Now the two type of structures can be formed. One is CH2OH, OH over here, COH, H, CHOH, CHOH, okay, CH2OH. Now the next structure that can be formed, COH, CH2OH, HOH, CH2OH. Okay. So how we can make this? This is fructose, this one, fructofuranose. Fructofuranose, you have furan ring, this is the furan ring. Fructofuranose, fructofuranose is this one. How you can make this? OH on the right side, right down. CH2OH, OH. Okay. OH on the upside, H on the downside. H on the upside, OH on the downside. CH2OH is always upward. This one is alpha. When OH is present on the right side, alpha. Alpha D minus fructose. Fructose. But remember, this negative is for the LIVO. Okay. This is alpha D minus fructofuranose. D is for the OH group that is present on the right side of this carbohydrate. Now, the next one is this one. Next one, how you can make this? This is your furanose structure. And here I am going to put up, down, OH on the upside, CH2OH on the downside. OH on the upside, H on the downside, H on the upside, OH on the downside, CH2, OH and H. Okay. So this is the structure, that is beta D minus fructofuranose. Fructofuranose. Okay. So this is the one, fructose. Alpha D minus fructofuranose and beta D minus fructofuranose. So these are the structure. There is some mistake, so I'm making it by myself. Okay. Now the next one is disaccharide, the linkage between the two monosaccharides by the glycosidic linkage. Glycosidic linkage is like, glycosidic linkage is very much important. Glycosidic linkage. Glycosidic linkage means oxygen linkage. Okay. So there is glycosidic linkage for the sucrose and maltose. Maltose are the disaccharides and sucrose is also a disaccharide. Sucrose is alpha D plus glucose, beta D minus fructose. Maltose is alpha D plus glucose, alpha D plus glucose. Okay. So this is for the glycosidic linkage in between. Now, next one is polysaccharide, large number of monosaccharides joined together, that is starch polymers of alpha glucose with two components, that is amylose and amylopectin. Here one twist is that amylose is water soluble, but amylopectin is water insoluble. Okay. Amylose is water soluble and amylopectin is water insoluble. This is for the starch. The cellulose, glycogen is the animal starch. Okay. Cellulose is plant based carbohydrate. Now the importance of the carbohydrates, they form the major portion of food. Like in potato there is carbohydrate, in you can say sucrose, means table sugar, also there is carbohydrate. It act as a storage molecule. Cellulose from the cell walls of bacteria, and the raw materials for the textile paper is also from the carbohydrate. They are a lot more important than these four. So these are some of the uses and the carbohydrates. Now, the next one is protein. Now, I'm going to discuss regarding protein. Now, protein, from the very beginning, you know that the protein are the building blocks of our body. Proteins are the building blocks of our body. Now, how protein will make? Protein will be synthesized from acids. Alpha amino acids, and L amino acids means left. How you can how I'll show you. So COH, the most oxidized in the top side. Here NH2 will be on the left side, H on the right side, and the R. R means any alkyl group. According to that, you will get alanine, valine, isoleucine, leucine. These are some of the amino acids. So, protein, how you will make the protein? Amino acids contain NH2 and COOH group. Amino and acid. Amino means NH2 and acids means COH. Proteins are the polymers of alpha amino acid. Okay. So how you can make, suppose I am taking, see all the amino acids, one topic is there, all the amino acids are optically active because they have chiral centers, because they have chiral centers, except except glycine, because the glycine structure is H. This one is glycine. Okay, so there is no chiral center, so they are not optical active. Glycine are not optically active. Now, how you can make a protein from here? Okay, suppose I am taking R H NH2 COOH. Okay, another R H COOH. So protein, after removing the water molecule from here, you will get a peptide linkage. A peptide linkage. This one is here, peptide linkage. Okay. Now the next one is, this is the peptide linkage. So this is dipeptide. Remember that peptide linkage doesn't make this dipeptide. Dipeptide is according to the number of amino acids that is joined. So this is amino acid joined together. Proteins are not made of these two, only these two. There are number of alpha amino acids that join together to form the protein. Okay. So I want to classify the proteins on the basis of the relative number of NH2 COOH groups. So for the neutral protein means there are equal number of NH2, means the acidic and the basic compound is same. Equal number of NH2 and COOH groups are there, then that is the neutral molecule. Okay, basic. Basic means more number of NH2 than the COOH group, and the acidic means more number of COOH than the NH2 group are there. Okay, this is the classification. Now the next one is the on the basis of place of synthesis. So there are essential amino acids and the non-essential amino acids. Essential amino acids that cannot be synthesized in the body and they need to be present in our diet. That is the essential amino acid. And the non-essential amino acid that can be synthesized in our by the body, and it need not to be provided from outside. Okay. Next one is on the basis of shape. Now, on the basis of shape, how we can explain? There are two types of protein, that is the fibrous protein and the globular protein. Fibrous protein is a fiber-like structure and the globular protein is the spherical structure. So there are a lot of things that like alpha sheets, beta pleated sheets, okay, alpha helical and beta pleated sheets. So there are quaternary structure, secondary structure. See, means in the starting peptide linkage is there. Okay, this is the primary structure of protein. Primary structure of protein means the peptide linkages are there. Only the amino acids connect in a straight chain. This is the primary structure. Again, the secondary structures. In the secondary structures, there are two types, alpha helical and beta pleated. Now, remember that as the hydrogen bonding and the interactions are going to increase, then there is the formation of the coil-like structure. After that, the tertiary, if the interactions are going to increase, like van der Waals forces, hydrophobic interactions are there, so the tertiary is more complex than the secondary one. Then the quaternary structure lot more, means the secondary, more tertiary structure combined together to form the quaternary structure. Quaternary structure. So after hydrolysis, suppose there is we are going to denature the protein by providing the high temperature or by changing the pH. But remember that denaturation means when the protein loses its native form. When a protein in its native form is subjected to physical change like temperature increase, pH changes, then the globules unfold and helical get uncoiled and protein lose its biological activity. Remember that the biological activity depends on the quaternary structure, but if we provide the pH or high temperature, then it will denature the protein, and because of that protein will not work like it will work in its native form. Now, so to combine this, after the denaturation, one thing that you need to remember is that denaturation doesn't uh, separate these molecules. Okay, it doesn't separate these molecules. Okay, so now the next one is next biomolecule. In the biomolecule, it is enzymes. Okay, enzymes are the proteins. Okay, enzymes are the proteins. Enzymes are very much specific, globular proteins specific for the protein particular reaction. This is important one, and for a particular substrate. Always remember, enzymes are very much specific. All proteins are enzymes, but all enzymes are not proteins. Okay, I am going to write this. All enzymes are proteins, but all proteins are not enzymes, because enzymes are very much specific. Okay, all proteins are not enzymes, but all enzymes are proteins. This is important one that you need to remember. Okay, next one is they are based on the lock and key model. Lock and key model. As already said, that enzymes are particular for the particular type of reaction. Okay, suppose as you know, each lock has its particular key. Okay, has its particular key that you are going to put and to open the lock. Each and every substrate has its particular structure, and an enzyme of that particular shape is going to enter in this substrate. Okay, like a lock and a key. You will get this one. This is enzyme substrate complex, and after that enzyme is going to uh, is not going to change the shape. It is going to form like this. Okay, again the same structure. This is your product. Mechanism. It reduces the magnitude of the activation energy. Enzyme function is like the catalyst function. Catalyst function. Okay, catalyst is going to function. What is the main idea for the catalyst using? To reduce the activation energy. Reduce the activation energy. Now, what is the activation energy? Activation energy is the energy required by the reactant to cross the energy barrier. Okay, so it helps the substrate to increase its energy or to lose the to reduce the activation energy to this point, so that easily reactant can cross this energy barrier. Okay, so this is the mechanism. Now the next one is hormones. Hormones is like chemical substance produced by ductless glands called endocrine glands. Okay, this is the hormones adrenaline and thyroxine. Adrenaline is a stress hormone that releases when you have a stress on yourself. Okay, so thyroxine, this is the hormones mainly not very much asked in the exam. Okay, next one is the vitamins. Vitamins are very much important, and it should be present in our diet. Organic compound required in diet in small amount to perform specific biological function for maintenance and growth. I told you all the carbohydrates, proteins, vitamins should be there in our diet too. Okay, to for maintaining our for the maintenance and the growth. Now the next one is classification on the basis of fat soluble and the water soluble vitamins. Fat soluble, now how you can remember this? Fat soluble and water soluble, just write it like this: A, B, C, D, E, K. These are the fat soluble. You just need to remember like this. Okay, A, B, C. B, C is in the water soluble. Water soluble means they excrete out from your body through water. Okay, by taking up the water. Fat soluble, that is in your body, that remains in your body, soluble in fats and oils but insoluble in water. B group and vitamin C are soluble in water. So this is about the vitamins. You need to remember some of the vitamins function, like vitamin A. The deficiency of vitamin A cause night blindness. Deficiency of vitamin B cause beriberi. Deficiency of vitamin C cause scurvy. These are the problems that is related to the deficiency of these vitamins. This you need to check. Okay, now the next one is the nucleic acid. Next one is nucleic acid. Suppose I am having this chromosome. Okay, chromosome, as already know that from the mother and father chromosome, a new child is uh, uh, going to get a birth. Okay, particles in the nucleus responsible for the hereditary chromosome are made up of protein and the nucleic acid. There are two type of nucleic acids, like deoxyribonucleic acid and the ribonucleic acid. In DNA, the main component is sugar, that is beta D2 deoxyribose, whereas in RNA, beta D ribose. Remember that 2 deoxyribose means the removal of a oxy group from the second position. Now, DNA contain ATGC, AGCT, or RNA contain AGCU. Always remember, uh, these are the main components of RNA. These are the nucleic bases. Means these are the bases. Nucleotides. What is nucleoside and what is nucleotide? I explain. Nucleoside. Nucleoside means which is formed from base sugar unit. Okay, there is no phosphate compound. See, base and sugar is the nucleoside. Base sugar phosphate unit, then it is nucleotide. Phosphate unit should be there. There are types of RNA, that is mRNA, messenger RNA, rRNA, and tRNA, transfer RNA. Now the biological functions, these are the chemical bases of hereditary, responsible for identity of different species of organism. DNA is the fingerprint of you. Okay, like fingertips are distinct for the distinct one. So this is the fingerprint of you. Nucleic acids are responsible for the protein synthesis in the cell. For the protein synthesis in the cell, nucleic acid is responsible. Now this is regarding the biomolecules topic. Now just quickly revise this. First, I am going to show one structure, one trick. Remember, get Rexel. This will help you in generating the structure. Okay. This one is glycerol aldehyde. Glycerol aldehyde. The structure of glycerol aldehyde only you need to remember. Glycerol aldehyde. Glycerol aldehyde is CHO, CHOH, CH2OH. This is a starting one. D is because it is on the right side. D is because it is on the right side. A most oxidized carbon should be present on the upside and the least one on the downside. Okay. Now this is for the glycerol aldehyde. If I want to generate E and D, E is erythrose, T is 3 O's. 3 O's. You just need to remember that you are going to form E and T. Again, CHO will be on the upside, CH2OH on the downside, CHOH. Okay. Here, E on the left side, then you need to put H on the left side, OH on the right side. For the 3 O's, it is on the right side, then you need to put H on the right side, OH on the left side. H, remember the H, where it is in this direction, T is in this direction, right direction. So hydrogen is going to be on the right direction. H, OH, CH2OH. This is erythrose. Remember this. When OH or the same group on the same side, then it is erythrose. OH, same group on the opposite side, then it is 3O's. Okay. Next one is ribose, a rabinose. XL. Xylose, lysose. Ribose, R on the right side, sorry, R on the left side, H on the left side, and OH on the right. This is just you need to increase the chain length. H, OH, CH, OH, CH, OH, CH2OH. Here CHO, COH, H, OH, CH2OH. Because H here on the right side. Now the next one is xylose. CHO on the left side, H on the left side, OH on the right side. H here, OH on the left side, same as it is. H, OH, CH2OH. Here the lysose will be on the H on the right side. Just you make this the same one. Okay. After that, if it is on the left side, H on the left side, increase the chain by putting H on the left side and OH on the right side. Here on the H on the right side and OH on the left side. I told you this one because I want to show you deoxyribose and 2-deoxyribose and RNA and DNA structure. This one is your ribose. So this one. Okay. Here you can see beta D 2-deoxyribose and beta D. you know that beta means if OH on the left side. Okay, I am putting OH on the left side because I have had already shown you regarding this structure, beta structure. Okay, OH, C, H, OH, C, H, here oxygen, CH2, OH. This is beta D ribose. Now, if I form furanose kind of structure, this is furan. Furan is 5-member ring. Here, this one is H on the upside. Sorry, right down. Right means down. OH, H. Again, OH, H. H, OH, CH2, OH. This is beta D ribose. You can see here beta D ribose. How you can form beta D 2-deoxyribose means DNA. This is RNA. To second position, this one is 1 carbon, 2, 3, 4. Second position, there is no oxy group. Here H, H. So this is 2-deoxyribose. H, OH, CH2, OH, and H. This is the important structure that you need to remember. Beta D 2-deoxyribose. So you can see beta D 2-deoxyribose, RNA is beta D ribose. Now the next one is, this is mRNA phosphate unit. After that, one another thing, get reaction I showed you. One another trick, all altros gladly make gun in giant tanks. This is aldos, altros, glucose, mannose, glucose, indose, galactose, talose. You can use the same trick. From ribose, you can make aldose altose, from arabinose, glucose mannose, from xylose, glucose and indose, from lactose, galactose and talose. On the right side, if from right side, H on the right side, if this is on the left side, H on the left side. Okay, just need to remember this trick to form the all the structure. And also remember that glucose and mannose are C2 epimer of each other. C2 epimer of each other. Okay, glucose and galactose are C4 epimer of each other. Epimers are the compounds which have changed in one position, in one configuration of the position. Okay, so this is the structures. With that, all the biomolecule is in on the tips of you. Okay, from the structures you got to know how to make this structure, you got to know how to know whether this is the straight chain structure, whether in the presence of carbonyl is there, whether the oxidation, this is the mild oxidizing group. So these are the structures that we need to remember. Okay, I hope I'm clear to you. Now the next one is almost covered, aldose, you can see all the structures here. Carbohydrates, then the second one is the classification, then the third one is the preparation we have discussed, fourth one is the how to detect the to get the evidence that whether the CHO and CHOH groups are present. This is the mainly, that's why I'm discussing it. Br2 water and HNO3 is a strong oxidizing agent. The difference, D plus glucose and D minus fructose. So these are the structures that you need to remember. Just remember the trick because this is the planar structure you are getting this one. Next one is ninth is this one. I told you how to make the structures. Write down, remember the trick, write down. Alpha is on the right, alpha when OH is on the right side. These are some of the important points. That is the anomeric carbon. If OH on the right side, OH on the left side, you will get this beta D minus fructofuranose. Glycosidic linkages, amylose and amylopectin. Primary structure, secondary structure, the lock and key model I have explained. Next one is the tricks, some of the tricks to give you an idea how to form the beta D ribose and beta D 2 deoxyribose. You can know how to form the structures. Sometimes they can ask the structures too. That's why I'm saying. I told you regarding the all altrose galadilimecan in giant tanks. These are the some of the important structures that I want to discuss. Thank you so much for watching.